Namaste students, I am JV Dr. Ajit Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic precipitation reactions. This topic belongs to the principles of immunology, basic examination, technology, and immunology. So precipitation reactions. In the previous videos, we discussed about the antigen antibody reactions, properties of antigen, and this uh, topic, precipitation reaction, is the type of antigen antibody reactions. There are two types of antigen antibody reactions: some precipitation reactions and agglutination reactions. So one by one, we are going to discuss precipitation reaction. What is the precipitation reaction? Precipitation reaction for an antigen antibody reactions between a soluble antigen and its antibody resulting in the formation of insoluble precipitator. The antibody causing precipitation is called the precipitation. So what is that? When an antigen and antibody which are specific to each other, and when they combine to each other, they interact to each other, they there is a formation of precipitate. They form precipitate. This precipitate is due to the antigen antibody and the reaction is called the precipitation reaction. And what is the mechanism of precipitation reaction? The precipitation is due to the formation of antigen antibody complex. First, it is antigen antibody complex formation. The antigen is multivalent here. Means it is multivalent. Means what is the meaning of the multivalent? For example, this is the antigen. For example, this is a antigen. Okay. So this is the antigen. This is the antigen. So this one antigen has 1, 2, 3, 4, 5 epitopes. There are 5 epitopes. 1, 2, 3, 4, 5. And the antibody must be bivalent. Means this antibody, this is the antibody, and bivalent. It can bind epitopes. Bivalent. It can bind to 4 epitopes. So it is a bivalent antibody. So antigen must be multivalent and antibody bivalent. As each antibody is bivalent molecule, it can bridge two multivalent antigen molecules. The bridge leads to formation of lattice, which forms the precipitate. What is the lattice? Lattice we are going to discuss here. And when antigen and antibody are in optimal concentration, the precipitation is complete. Large lattice is formed. So the lattice is a condition when concentration and antibody concentration are equal. Okay, and when they are equal, they form a precipitate or lattice, which is considered the precipitation reaction is complete. So when a soluble antigen combines with its antibody in the presence of an electrolyte, means NaCl, at a particular temperature and pH, it forms an insoluble precipitate of antigen and antibody complex. Means in a beaker, we are adding antibodies, and in another mixture, we are adding antigen. When we mix both of the mixtures, then they form a antigen antibody complex. complex. So what are the types? What are the media which are used in the precipitation reactions? So there are precipitation reactions conducted in two media. The first is liquid or in the gel. First, we are going to discuss precipitation in the first liquid. What we do in the liquid precipitation, a set of five or more reaction tubes are arranged serially. One, two, three, four, five. Five tubes are arranged A, B, C, D, and a constant volume of antiserum is added. So suppose that we have added a constant volume here in the antibody. The antigen is then added in increasing volume. When we add antigens, suppose we are adding one antigen, 1 ml, 2 ml, 3 ml, 4 ml. So in the increasing order, and in all the five tubes, antibody strips, but antigen is added in the increasing ratio. Means 1 ml, 2 ml, 3 ml, 4 ml, 5 ml. The amount of precipitate formed, then after that the antigen and antibody react to each other and they make a precipitation. They make a precipitate, and the amount of precipitation formed is determined by the proportion of antigen and the antibody. When the amount of precipitate is formed in the different loops is plotted on a graph paper, then a curve is obtained. So I can share the screen here so that students can understand the diagram better. Well. So this is a diagram. Here there are five tubes, one A, B, C, D, E, in which we have added antibodies in equal proportion. For example, we have added 2 ml or 3 ml of antibodies here, and we are adding antigens in uh, increasing proportion. For example, 1 ml, 2 ml, 3 ml, 4 ml, and 5 ml. In the medium tube, we are observing that the precipitate is formed here, which indicate that antigen and antibody are in equal proportion. Here, antigen and antibody are not equal proportion. Antigen and antibody reaction is formed, but precipitate is not formed. There is not any equal proportion of antigen and antibody. So when the antigen and antibody proportion is equal, then we can find that that is the concentration of the particular. So here in the C2, we can find here that the precipitate has been this is the precipitate. And these are the antigens and the antibodies have been attached to that particular antigen. So when we plot it out, we observe that there are three zones are formed. Three zones. In the first tube, there is a low level of antigens. So antibodies high in concentration. Number 1 and 2, the concentration of antibody is higher. So it has zone of antibody excess. It's found in the antibody excess region. Sorry. In the third number tube here, and in the 4 and 5 number tube, the antigen concentration is high. So it is showing the zone of antigen excess. Zone antigen are in higher concentration, and in the tube number 1 and 2, the concentration of antibody is higher. So the two region, zone of antibody excess and zone of antigen axis are there. What is third number two? Where a precipitate is formed due to the concept that antigen and antibody concentration is equal. So it is a zone where the antigen and antibody concentration is equal, and it will be considered as the zone of equivalence. Okay. So three zones are there: zone of antibody axis, zone of equivalence, and zone of antigen axis. Now, precipitation is in gel. Uh, it is a procedure in which we conduct a process in the gel, agar gel. Both antigen and antibody are added in agar gel. Antigen and antibody diffuse freely in the system in all directions. At a certain point, the rate of diffusion and the concentration of reactant are equal, and the precipitation is formed, which is which can be easily observed. This is the radial immunodiffusion. That is the all directional. Uh, in this, we added antigen and antibody. That is the radial. We added antigen and antibody, and these antigens will move, and antibody will also move. They will diffuse at the end, where antigen and antibody will move at the corners where the concentration of antigen and antibody is equal. They will form precipitate, which is known as the precipitation. Also, we can find here that they are in equal proportion here, and the remaining antibody, remaining antigen are here, and remaining antibodies are here. But at this area, the antigen concentration and antibody concentration is equal. Similarly, the double immunodiffusion can be performed also. In the double immunodiffusion, what we do, we have a gel system. In this gel system, uh, we separate the antigen and antibody zones. In single area, we add antibody, and in another portion, we add antigen. In the gel, the antibody will move, they will diffuse from high to low concentration. And similarly, the antigen will also move from the high to low concentration. So at the middle portion, the concentration of antigen and concentration of antibody will be equal. They will move to each other, and they will form a antigen antibody complex in forming lattice. So after formation of this lattice, the antigen antibodies will form a precipitate, which is known as the precipitation reaction. So by understanding this, we can understand what is the precipitation reaction. So by this lecture, students can be able to understand what is the precipitation reactions, what are the types of the precipitation reactions, and what are the media which are used in the precipitation reactions. Students can also understand the examples of precipitation reactions and experiment related to the precipitation reactions. And what kind of questions can be asked in precipitation reaction? The questions which can be asked in precipitation reaction is that explain antigen antibody interaction. Explain example of antigen antibody reaction. So we can answer with precipitation reaction. If somebody asks that, give any example of the give any example of the antigen antibody reaction, we can answer with the precipitation reaction. If somebody asks about the precipitation reaction, then we can directly answer it. It can be also that explain the precipitation reactions with suitable examples. Then we can answer it with the experiment, 5 tubes in the liquid and in the jar. If somebody asks about the question, explain antibody interaction with suitable examples and explain and precipitation reaction with with experiment, and which experiment can be conducted. Two experiments are conducted in the precipitation reaction. The first one is autolony double diffusion assay, and the second one is radial immunodiffusion. Two experiments. So my lecture is over here. If students have any questions, they can ask me directly. And, you can ask me in the YouTube channel. Thank you very much.